This lecture is on the small intestine. The small intestine, as you recall, is bounded by the pyloric sphincter, which leads out of the stomach. So, the gross anatomy then of the small intestine is that it measures approximately 6 meters. That is to say, approximately 20 feet then, in length, and the diameter ranges from about 4 centimeters at the stomach, then to 2.5 centimeters at the junction with the large intestine. So a little bit of variability in diameter in the small intestine, but still altogether narrower than the large intestine. That, by the way, is why it's called the small intestine. It is not shorter than the large intestine, but it is narrower than the large intestine. So the small intestine then is subdivided in, basically for our own purposes then, into 3 segments. And they then are the duodenum, which is the part nearest to the stomach. Some people will call it the duodenum, usually called the duodenum. The next is the jejunum, the middle section, and then finally the last section is known as the ileum. The ileum is what has the boundary then with the large intestine. So we're going to start just by talking about the duodenum. So the duodenum then receives chyme from the stomach. And it also receives secretions from the pancreas. That would be all those digestive enzymes, and also the liver, where it will be getting bile. And the duodenum then makes up the first 25 centimeters, that is to say approximately 1 foot then of the small intestine. So it is therefore about the first foot nearest to the stomach. And then there is a rather abrupt bend. You can see this in diagrams and in dissection if you do one then. So there's an abrupt bend in the small intestine then that marks the boundary between the duodenum, the first part of the small intestine, and the jejunum, that second part. Now, the boundary is really just something that we note. There's no sphincter there or anything like that. It's a continuous organ. It's just kind of our way of understanding and talking about the small intestine. So that bend then is where we stop talking about the duodenum and start talking about the jejunum. So then we have the jejunum. And this is, for the time being, all I'm going to really say is that this is the next 2.5, 2.5 meters in humans. That is to say, about 8 feet then of the small intestine. And it then leads to, of course, the ileum. And the ileum is the last portion of the small intestine. And all of these sections are well anchored then in the peritoneal cavity by membranes that are called mesenteries. So mesenteries are clear membranes. If you dissected a frog in high school or anything like that, you may have noticed this membrane that was grabbing onto the small intestine. A lot of people just kind of pull it apart in order to see the whole length of the small intestine. But those membranes are incredibly important. Those are the mesenteries then. And what they do is that these then attach the small intestine to the dorsal body wall. Now you remember that dorsal means towards the back. So what this is saying is that you have all of these guts. You have this small intestine, which could just slosh around on your insides, but instead it has this clear membrane attached to its entire length, and then the membrane kind of bunches up and winds up attaching to your dorsal body wall. So basically towards the interior of the abdominal cavity at the back. And that is what holds your small intestine kind of in its place. It is not, it's not rigidly attached, but it is firmly attached there. The mesenteries also contain blood vessels. Those are the blood vessels then that go to the absorptive areas of your small intestine. So those mesenteric blood vessels are incredibly important. So that then is the external anatomy of your small intestine. Now let's talk about the internal. Anatomy of the small intestine. First, there are transverse folds in the inner surface. So in the mucosa, remember that's the mucous membrane lining your small intestine. So we have these transverse folds in the mucous membrane then of the small intestine. And these then are called plicae. So what did I just say? I said basically here you have a tube, okay? I've sliced through a tube. Here's the outer wall of the tube. And then on the inner lining of the tube, we have these folds. I'm overdrawing it here. But the point is you have these folds, and they then are the plicae. And the plicae are a permanent structure. So they just stay there. And these increase surface area then for absorption. In this course, you're going to learn to think like a physiologist. And when you see folds or any kind of sort of, you can't quite figure out why there is all of this seemingly unnecessary tissue and unnecessary in sort of ornateness to the tissue. Very often is to increase surface area. And that's what's happening with the plicae, because we need that surface area for absorption of nutrients. Space is limited inside our abdominal cavity, so we have to make the best use of it that we can. So as a result, we have all of these little structures that allow for more surface area in our small intestine for absorptive purposes. So, as I said, we have the plicae there to help with that. And then we get more fine structure here. Each plica, that's the singular of plicae then, each plica supports a series of finger-like structures called intestinal villi. So what do I mean by that? We have these projections. I could easily, instead of structure, have said projections, but here's what I'm getting at. Here we are going to look at a cross section of the small intestine. There's the outer wall. Now, we've already talked about plica, plicae. But now, on each one of these plicae, what you are then going to see is these finger-like projections that go into the open area of the small intestine. So you see here's one, and here's one, and here's one, and here's one. So these things here that push their way into, that project into the small intestine, then these, and I'm not going to label the other two, but there they are, those are your intestinal villi. We're still not done. So those are your villi then. And then each villus, which is the singular of villi then, each villus is carpeted by what are called microvilli. And so these then are small or even tiny, almost hair-like, very short hair-like projections. So let's do this out a little bit more nicely this time. So here we have a cross section through your small intestine. Let's draw in the villi now. So there are the villi, and I'm going to make the villi in blue just so you can see them a little better. So here's a villus. Here's a villus. Here's a villus. And here's a villus. So these are then projecting into the opening in the small intestine. The opening is known as the lumen, just so you know. But then, projecting off of the villi are teeny little hair-like things. It winds up looking sort of like velvet or like a carpet that has really short fibers sticking out. I'm not going to draw all of them, but you get the idea then. These tiny hair-like projections then, these are your microvilli. And again, you can see how this is going to greatly increase the surface area inside your small intestine. Okay, so we have these microvilli then. And if the small intestine were a simple tube with smooth walls, then it would have a total absorptive area then of approximately then 3,300 square centimeters. Seems like a lot. Seems like a good amount, but not good enough. So then, by contrast, with the plicae, villi, and the microvilli, the total absorptive area instead is approximately 2 million square centimeters. So it's a huge difference in absorptive area just by having all of those projections. Now, a few other things that you need to know then about the interior of the small intestine. We have intestinal cells there, of course. So the intestinal cells then are exfoliated, basically sloughed off. On a regular basis. They live, they die, they get sloughed off. And these then simply go through the GI tract and are excreted. Basically, they just follow along with the chyme that's in the GI tract and wind up being excreted. Now, just for a little bit of sophistication in your outlook, a little bit of detail as to where we find the villi and the microvilli and plicae and so on, where we find them in abundance and where we do not, that depends a bit on what part of the small intestine you're looking at. So, the plicae and villi then, first of all, plicae are absent near the stomach and the villi are short. And then further along the duodenum, the plicae and villi become more prominent. You may begin to ask yourselves, why? And then these achieve maximum size at the start of the jejunum. So what's going on with this? It makes sense when you think about it. So we have the chyme coming out of the stomach. It's highly acidic. Why would you want a lot of surface area exposed to acid? On the other hand, as the nutrients in the chyme get broken down into absorbable units, then you need to kick in the architecture that has to do with absorption. So basically, because chyme becomes less acidic as it goes along. So it's going to damage the plicae and the villi and so on less as it goes along. And as I said, you need the structures for absorption more as you get absorbable units. Hence, carrying on from there then, hence you also need fewer mucus cells because mucus secretions protect the intestinal lining. And as a result, more surface area can be supported. And then on top of that, this is also where you really need more surface area for absorption. So that then is our lecture on the small intestine.